So basically, nature journaling is, this is the way I see it. It's observing nature and it's recording what you see. And you're using a combination of words and numbers and pictures. That's what it is in a nutshell. And when you're doing nature journaling, you are practicing how to observe nature more closely, honing your observational and awareness skills, practicing your drawing skills, practicing how to put visual data or data into your notebook. And also, another good news is you don't have to be an artist and you don't have to be a scientist or a naturalist. Right? You can have no experience in all of that stuff and you can learn all of it with practice. So if you're new and you're a little unsure about your skills about anything, not to worry. We're going to walk through everything with you. So today might go a little bit fast, but don't worry. Some of the classes, some of the Sunday sessions are a little bit longer and we do a little bit of a lesson in the beginning. So I'm just going to do an overview here. So observations, we can ask, there are 3 sentence starters that help us out. So you can write in your notebook right now, you can put I notice. And these are really great prompts to help us think about what you want to put in your nature journal. So here's, I'm going to show some examples of other people's pages where they've drawn some pictures, put some numbers, put some words to show you what a nature journal page can look like. And our focus with nature journaling is not making a pretty picture. It's not art, okay? So don't worry about making it artful and beautiful and, you know, super realistic. It can be sketches, as you can see in this picture. You know, you just draw what you see, like this hummingbird. You can't really see every feather on the hummingbird when it's flying in front of you, so you draw what you see. All right, so I notice is the observation portion. And then tapping into your curiosity, you start to ask, I wonder. You know, I wonder why this bird has a strange bill. I wonder why this fish has this weird shape. I wonder what he does with this tail. All of these questions you write down in your nature journal. Actually say them out loud. Those are really great when we're out in the field and we see stuff. Say it out loud. It's going to help you remember those questions and then write them down as soon as they come up. Because I guarantee if you don't write it down, it's going to disappear. And I think that nature is always speaking to us when it catches our attention and a question pops up. And by capturing it on the page, it gives us an opportunity to get to know it better. And oh, one thing about questions. So with these questions, they're not meant to be answered. So I want to just preface that. So we're putting questions down, all these questions I said, like, why are these fish schooling together? Why are all the same size? Why do they swim so close together? Why does that bird have that bill? How does it eat? Does it eat, you know, little things? Does it dig in the sand? Does it, you know, pick off the ground? Does it, you know, lift rocks? All of these questions put down in your nature journal, because that is going to focus your observation. And right, remember I said nature journaling is about observing and recording nature. So all of these questions are going to help you observe more deeply, so that sometimes these questions get answered just from your observations. And that's pretty exciting, right? To have the question and then find out the answer on your own. So sometimes on these calls, you might have a question and it might not answer. And it's not that I don't want to tell you the answer. It's because I want to encourage you to sit with the question. So I want everyone to be comfortable with not answering questions, right? And at the end of the session, if you're really curious about one question, then by all means, you know, go and explore some more, but observe as much as you can to get that answer or question answered on your own. And then, you know, delve into it deeper and follow your curiosity, whatever you're interested in. So again, lots of questions, write them in your notebook. They're meant to be in your notebook. And by putting words on your page, the pictures lose their power to, the inner critic loses the power, I should say, to judge ourselves on how good that drawing is. So put lots of words on the page. And so here's an example here. This person put lots of words, labels and titles and information and names, questions. And then the third part of this triad is connecting. So we start with a sentence starter, it reminds me of. And that just gets us kind of, you know, getting our creative juices flowing. We start to think about, what is this thing that's in front of me? What does it remind me of? And it might remind you of something that's non-natural, or it might remind you of a memory, or a smell or a childhood experience. So I encourage you to write all of those down, because when we make those connections with something that's outside of what we're looking at, we deepen this understanding and awareness. All right, so those are the three, the triad, the I notice, I wonder, it reminds me of. So those are really great sentence starters to put in your nature journal. All right, and then what do we record in our journal? We use a combination of things. Sometimes words show something better than a picture, and sometimes vice versa. So we'll use a combination of words in the form of titles, labels, pictures, and diagrams. You'll see these crows here. They're not super detailed. I didn't put like every wing or the feathers or even the eye, because what I saw, what I wanted to capture in this drawing is not the details about the crows, but the fact that they were collecting these grasses. So just think about what you want to show on that page, and draw a diagram of what it's doing. And sometimes, that'll take the pressure off of, oh, it's got to look like, it really needs to look like a crow, or it needs to look like, a petunia. In numbers, we had a session a few weeks ago about numbers. We'll talk more about numbers next week, but basically just count things and measure things. Putting temperatures and the dates, those are all numbers. So, these three languages combined together will make a really rich page. And so the goal with nature journaling is not the final page, but it's really the process. So we want to observe nature more deeply and learn how to learn strategies on visual thinking. So that's basically what it is. And if the page turns out to be beautiful, that's great, but I want that not to be the goal. Right? So that way, anyone who's got any skill level can do a nature journal page. All right. Okay, so we're going to jump in. So today we're going to be looking at some plankton that's alive and living, and they move. So they're not going to be holding still for a pose for us to draw. So we're going to do some warm-up sketches, and I'm going to share my screen with you. We've visited this website before. It's, oops. Oh. line-of-action.com, and you can sign up and take a look at what they have to offer. But they have this really great feature where you can do drawing, and they'll run through a whole bunch of random pictures, and you can set the timer. So I've set the timer for 60 seconds. I made it a little bit longer today than we normally do, just so that you're feeling comfortable and warming up. So first of all, the purpose is not to draw the butterfly, okay? We see a butterfly, but the purpose is to look at the general shapes. So maybe what I'll do is I'm gonna share my, my video so that you can see what I'm doing. All right, so with gesture sketching, we are, so basically what we're doing is we're looking for general shapes. So when I'm just going to start sketching, and I'm going to talk it through, and you do the same thing. You don't have to copy what I'm doing, but I'm just doing it here so that you can get a sense of how much detail I'm putting into this. So I'm going to play this, and this is 60 seconds. So it's short enough that we're not going to do the whole thing. So I see oval for the body and these kind of big triangles. Just block them out and use a light touch with your pencil, and lots of like light sketchy lines. And it doesn't need to be perfect, because we're just warming up. We're getting our hand and eye working together. So don't worry about the details and the stripes. We're just going to get the general shape, and then I'm going to maybe refine it a little bit since we have some time in the 60 seconds, and maybe get a little bit more of carve the shape out a little. And then the body and the little antenna. And basically that's all we need to do. We don't have to get all of these details in, because these are gesture sketches. All right, next one. Okay, so I see I'm gonna do like a circle for his head, big kind of a square for the ear, and the trunk comes out, big kind of ovalish body. And you can see I'm just using light sketches and just eyeballing where I see this body shape. And then the leg. This might be a little hard for you. It's okay. Hang in there, guys. If this is challenging, just do the best you can. Light sketches. I promise that after a few sketches, your eye and your hand is going to warm up and it's going to get a little bit easier. All right, so the goal is not to make it look like it doesn't have to be the elephant. You're just blocking out the general shapes. And don't worry about details like the wrinkles in the eyes. We're going to get the big shapes. And then if you have some time, you can fine tune the shapes, and you notice the shape of his feet. Okay, let's move on to the next one. All right, some general shapes. I'm seeing the big body oval and oval for the head. And see, we're practicing getting proportions so that we're not making him too big and too small. His leg comes straight down. All right, those of you who are just jumping in, we are practicing some gesture sketches. So grab your journal and a pencil. We're just making light, sketchy lines, and we're just getting the general shape of each of these animals. Don't worry about getting any detail. This is a warm-up to get our hand and eye working together so that when we get the plankton that's moving around, it'll be easier to draw. All right, so general shapes. So I'm seeing like a circle for the head, and the muzzle's kind of like a cone. And the neck comes down. Just blocking in these like basic geometric shapes like ovals and squares. And just a really light touch on your pencil. You don't have to hold it firmly, and make a bunch of light lines. Yeah, just general shapes. And it's okay if your drawing looks like a snowman and not like a cat. It's just getting those basic shapes down. And for those of you who don't already know, drawing is not a talent. And if you've never drawn a bee before, it's totally okay. You can block this out. It's all about just practicing drawing what you see. And if you've never drawn what you've seen, then it might be a little challenging, but the more practice, the better your drawings will be. Because your hand and eye need to learn how to work together. And if you haven't practiced how to draw what you see, then you need to strengthen those muscles, so to speak, those connections. Oh, see, I'm going into detail already. It's really hard. It's like you got to fight the detail and just get the general shapes, right? Like this. There we go. I don't need to put all the hairs in. Okay. So drawing is a skill, a skill, which means that anyone can improve with deliberate, intentional practice. And one thing that John Muir Laws has coined of pencil miles is the idea that the more miles you put on with your pencil, like the more that you draw, the better your skills are going to be. You're going to improve. So even doing these gesture sketches, like even if you're not drawing something from nature, you're just doing some quick sketches and practicing, this counts for improving your ability to draw. So, I mean, I'd encourage you to, if you can't get outside or if you feel like you don't have anything to nature journal, you can go to this website or just go to the internet and just set a timer and just go for 30 seconds or 60 seconds and just sketch. And the beauty about these quick sketches is that you don't have time to finish. So it can't be a finished drawing. So it kind of tricks our mind, right? Okay, how are you guys feeling? Hopefully you're kind of starting to get into the groove, starting to see. So we're training our eye to see shapes instead of the details. And hopefully you're warming up. Right, so it's not about a finished drawing, so don't worry about putting any detail in it. And then now you can walk away from your sketches, you can turn your page, and we're gonna move to a new one because we are gonna welcome Janai from PacificPlankton. I think that was my slide, and I'm gonna stop my share so that Janai can come on.
Hello. All right. Well, welcome everybody. So one of the cool things about that warm up is that we're viewing Richard Parsons' screen.
Uh-oh, how did that happen?
So a couple quick things. I am not a marine biologist. I'm not a diatomist. I do not research these creatures for an organization. I fell in love with them, and I came to this through an art background. And a few years ago, I started photographing what I was seeing through the microscope while I was volunteering at the Greater Farallones National Marine Sanctuary's Visitor Center in San Francisco. And an awesome team of people there let me use their microscope, and they're like, yeah, sure, you can hook up what you want to that. And then they're like, oh, do you want a monitor so you can share with people at the visitor center? I was like, yeah. So I started manning a plankton station at their visitor center 2 days a week and sharing what I found. And I started this with all of the questions that Melinda shared and that Jack always asks. The I wonder was where I started. And the more I saw, the more I wondered. I've since then started taking classes at the local community college. So environmental science and oceanography, ecology, biology, to try and deepen and broaden my understanding because I wanted to go deeper. So today we're not going to be able to answer everyone's questions. If you have questions though, put them in chat. And if you do work in one of these areas and you happen to have the answer, feel free to add it in chat if you'd like. But I think the questions are really important because this is for me, it was a completely alien world, and it's just amazing. So I'm going to share my screen now. Let's see, and give you a little bit. Here we go. Oops, I need to go back one. Nope, that's where I'm starting. Nope, my computer, there we go. So the Greater Farallones National Marine Sanctuary is the organization, and the Farallones Association, Greater Farallones Association is the nonprofit that supports them that I started volunteering with. I think you can see my mouse way down here in San Francisco Bay. Yes. Right there's the little visitor center that I was volunteering at. And so let's see if I did this right. It should be able to. Ha, there we go. The little red and white pier is where I would usually collect plankton, but because of the pandemic, it's not happening right now. There's a classroom at the end of the pier. So what I would do is I would take my plankton net or their plankton net, and I would walk it back and forth up and down this pier. But because of the pandemic, that's closed. So yesterday, at the end of the day, I took a plankton net with my mask and I social distanced from all the fishermen on the Torpedo Wharf, which is near the warming hut in San Francisco. So if you wanted to know more about those particular places, you can look them up. And then here, because we've been having bioluminescence recently, I believe down in Monterey, there's also bioluminescence in Tamales Bay. And this was a nighttime kayaking trip that we went on to go check out the bioluminescence there. And that's pretty amazing. Let's see, here we go. So one of the things for nature journaling is kind of the numbers piece. And the numbers piece for plankton is information about the environment, not just the day, but the water temperature, the air temperature, the whether the tide is coming in or going out. And so at the end of that little red and white pier, there's this building here. And that building houses all of this really amazing equipment that is constantly collecting that data. And so if you're interested in starting your journal page, if you're wanting to, this is the actual data from the day and the time that I collected the sample. And I'm going to leave it up for a couple of minutes. I'm just going to walk through a little bit of it. So this was collected yesterday, and it was collected at approximately 6 p.m. So you should have information from 2 different organizations, but it was all collected at the same location.
So Janai, that
So
was the 29th. So we could put August 29th.
Correct.
So
Today's
these are great numbers. Yeah. So August 29th at 6 p.m.
Yep.
Okay.
And so last update for the Bodega observing node, Saturday, August 29th, 6 p.m. Pacific Daylight Time. So briefly, while you're copying numbers down, if you are capturing this.
It's kind of, it might be kind of hard to see the number. So maybe you can just tell us what the seawater and air temperature was. Maybe those are just some basic stats that we could put on here.
Yeah. Sure thing. So the tide was incoming. So it was going from a low to a high tide, and it was 1.271 meters at the time. I try and do, you can put it into a conversion thing if you want, but I like the metric measurements personally. The water temperature was 16.8 degrees centigrade. And I found that was interesting. I was wondering about that because the air temperature was colder. It was 13.9 degrees centigrade. So that's something that I look at when I'm doing the difference between the air and the water temperature. There's a lot more information that you can deep dive into at the websites for the data collection, but those are the water level, the water temperature, the air temperature are the things that I'll often pay attention to. And then sometimes if it's been raining a lot, I might look at the seawater salinity, which is over on the Bodega Bay observing node. And so 26.753 is how that would be recorded, PSU. And I know that they changed that. It's like, it used to be parts per thousand, and I can't remember what the new universal unit is, which is basically the same. If somebody knows in chat, please feel free to pop it in there. So this is how I collect the plankton. I use a net, and this is what the net looks like. It has a really fine mesh. At the end, there's a jar that you use that collects the goodness. So think of it kind of like a colander to catch the really small stuff in the water. So by dragging it through the water flows out through this super fine mesh, and all the goodness gets collected and washed down into this little end. This here up above is the actual net that I used. This one, it's got a fine mesh. It's approximately a 60 micron mesh. So I didn't tow with, I have 2 nets that I sometimes have access to. I only had access to this one net because of the pandemic. And my sample was not as golden brown as that one, but it was definitely more golden brown than what I would have gotten if I had gotten, used a net with bigger holes. I captured more of the phytoplankton. And these 2 websites, I can add in chat later, but they're where the data came from. And so the last bit is, this is my science fort. This is where I hang out and do my, my nature journaling and my observations on plankton in my house. I don't have an awesome lab with stuff, but I have a microscope. This is my microscope. This is the camera you're seeing everything through right now. It's a Canon camera. And down here, this is a dark field filter. So this is the part of it that I'll be using today that will block the light coming straight through the bottom of the microscope, so that what we see is illuminated on a dark background. And I'm gonna bop over to, let's see, here we go, there's some plankton. And I'm gonna see if I can, too many, oops, nope. There we go. I was gonna try and open up my, so that you can see if I do any drawings here. So while you're getting a chance to just peek at this, I'm gonna try and, oop, there we go. So there's a lot of beautiful golden brown stuff. And I am noticing some flashing, sorry about that. I've got a connection that's loose. If it keeps it up, I'll have to readjust. But what I'm noticing right now is I'm noticing there's a lot of round, let's see if you can see if I draw here. A lot of really round, golden brown. No, it's too bright. Let's see. No! Why is it flittering?
It was doing pretty well for a while for us. We can see it right now.
It's good now.
It's good right now. All right, I'm gonna try and.
And folks, while she's adjusting this for you, if you would like to see the view a little bit differently, at the top of your screen, if you choose view options, you'll see that you have a fit to window and different percentages. I prefer side-by-side mode, so you might want to try that if you're not on it. And that should allow you to see things at different sizes so that you can see the plankton and the drawing and Julia.
All right. So if it keeps flashing like this, it might be that I'm overwhelming my computer, and I might need to switch to a different viewing mode. So for right now, we're gonna try it like this. So we're gonna walk through. First of all, what is plankton? You need to be 3 things to be plankton. So in order to be plankton, you need to be alive, you need to be in the water, and you need to be a drifter. And by drifter, I mean, you can swim around. A lot of these organisms are moving around, but they're not able to swim stronger than a current. So if the current is going from San Francisco to Los Angeles, they're along for the ride. They aren't going to be able to change direction and decide to swim to Hawaii. So they can manage where they are in the water column, but they get pulled in whatever direction the water's flowing. But right now, I am noticing there's a lot of these golden round circles. So those are phytoplankton. They're a centric diatom. And even though it only looks like there's one, if we look carefully, we can actually see that there are more than just one. I'm gonna switch out because it seems like my computer is struggling. So let me switch out. If I stop the share and I go here, let's see if this is any better for everybody. There we go, maybe. And so hopefully hopefully it'll improve. And I'm going to quit that other application I was using in the background because it's eating up my, there we go.
So can I just jump in here? While we're nature journaling, folks, just when things are moving by quickly, you start to draw something in one position, and then just if it moves away, then start another picture. So I was showing my screen earlier about how I'm drawing the plankton that's moving really quickly, and it's the same way that when we're drawing birds or if we're drawing squirrels, you're going to start with a quick sketch, adjust your sketch of maybe one of these polychaete worms, right? And then when it moves out of the view, then move to another spot and then draw a different position, whatever position it's in, and then as it comes back to that another position, you can fine tune and add more detail.
Thank you. So here's another thing. So this is a really heavy sample that I put on to begin with. And I like to get a really rich sample on a slide at the beginning of my microscope session. It gives me a good impression. It'd be like if you arrived at a lake or water wetland and you looked around to see what was there, and you might do a landscapito and be like, oh, I see, like for me, I'm seeing a lot of these golden brown circles, and so that's a big thing for me to notice. I'm noticing, and I'm wondering about everything that's moving really quickly. I can't really tell what they all are yet, but just seeing them moving around, each of them moves in a different way. And so I wonder about there are some that move in quick straight lines, some that are more sinuous, and in their movement, I notice that I'll notice there are some that kind of flop or flap. And I relate this a lot to bird watching because for me, watching the plankton is kind of like watching a lot of birds in a wetland. You can't always just pick out one. Sometimes you have to see them as a group. And it's the way they move that gives me clues as to who they might be. So I'm going to move around this slide. I'm going to take a couple minutes here, and I'm just going to scan around. So don't get frustrated and try and keep up really fast. Just try and get an impression of what's available here in the plankton today. And then I'm going to do another slide after this. And I'm going to get some phytoplankton on it. So the phytoplankton is the plant plankton. It's the usually golden brown or green component, and they make their food from the sun. And there's so much diversity and variety in it. It's pretty amazing. So here, I'm just going to scootle around the microscope here just to get an idea. I know we were holding still in one place.
You know, I'll just encourage you, everyone, not to worry about like what everything is right now. Just like what Janai was saying, just imagine this is a flock of birds and we're watching them, and write down what you notice. Like I notice that there's green circles that are still. I notice, and you can say these out loud to yourself too as you write them down. I notice a lot of green circles that aren't moving. I notice some white circles that aren't moving. I notice there's some wormy things that are squiggling around really quickly. I notice that there's smaller little creatures that have a shape that look like a chalice that's wiggling around. So, you know, all of these things, just write down. You don't have to know the names of what you're seeing. Write down these words of what you notice, and then just sketch what you can see. And you won't be able to get a perfect picture of any of this plankton, and that's totally okay. And like Janai was saying, she's gonna get a different sample, and we'll be able to see some of the phytoplankton, which is the plant plankton, a little bit later. But I mean, just look at the amazing diversity of what you see. You know, like these different shapes, and just sketch them out.
So I'm having trouble. Is somebody is it my video that's dropping off because it keeps
It looks like you keep coming on and off of the feed.
Okay. All right. Hopefully I fixed it. There we go. Because my computer seems to be having a little bit easier time now. There we
Oh,
go.
and Janai, would you tell us all again what the field is? Was it like 40?
Yeah, so right now we're looking at this. If you were looking through the microscope with me, through the eyepiece, you would be seeing this magnified 40 times actual size. And that's because I'm using an objective, which is the part of the microscope that points down by the sample. That's the objective, and that's a 4 times objective. And then the eyepiece on my microscope is a 10 times eyepiece, and the eyepiece is where I place my eye up to see. So you multiply the 4 times objective by the 10 times eyepiece, and you end up with 40 times magnified. And because everyone's watching on different screens, what that means for you is from the width of your screen, whatever it is approximately 5.5 millimeters at this magnification. So all of this that's happening is happening in a width of about 5.5 millimeters, so half a centimeter. I'm gonna scoot around a little bit more. So there's some stuff that holds still and there's some stuff that moves. And the more you observe, let's see, the more you start to see, there's a difference in color. So there's like the more golden stuff tends to hold still, and the clearer stuff, here's a really dense section. So this settled out towards the center of my slide. I'm gonna kind of scoot past it.
Janai, how many drops of water is in this slide that we're taking a safari through?
Good question. So this particular slide is it's a deep well slide, and it holds from between 15 and 20 drops. Ooh, there's a polychaete worm, that worm that's just swimming down. Cool. And a lot of the other little worms that we've been seeing go through here are polychaetes. So it's a type of worm. They have spines. We might not be able to see it. They're really beautiful. And there's quite a few of them.
And so these are good things to write in your journal, that how is it wiggling? How is it moving? To me, it looks like it's like, a worm that's moving through grains of sand.
Yeah. Oops. Let me see if I can go back this way.
And we have a question about the rocks that are in the view. Someone is wondering whether that's what that is. Is that sand?
Yes. So the things that look like little rocks, they're little rocks. They're because I collect near shore, right? You saw where that red and white pier is. It's not way out in the middle of the bay. It's not way out in the ocean. I'll get things that are stirred up from the shoreline. So everything from bits of plastic to little bits of sand and rock, but they're very tiny. I mean, these would be the super fine. These aren't giant coarse grains. Here, to give you guys an idea, I think this will help. I have a tool that is like a needle that I use for poking things around, and I'm going to put it on the slide. I'm going to slide it into the field of view. Let's see here. Here it comes. I'm going to try and get it. You can see that point. That is basically, oops, which way? There we go. The point of the needle. It's not a super fine needle, and it's not a giant chunky needle. It's just kind of your generic average needle. Just to give you an idea of the size difference with something that you might be more familiar with. It's probably close, a little bit like the tip of a push pin maybe. But that's how small the things are that we're observing right now. I'm just going to hold it there for a second in case people are interested in just getting an idea of the perspective on all of this. So I'm I'm looking at the stuff, and I just noticed that there's a different shaped organism. It looks kind of like it's right near the point right now.
Mm-hmm.
It's kind of a star shape with fine points, and there's a ball at the center of it, and it's just getting bumped away. It's like they're playing, they're playing soccer with it.
Colored.
It's kind of a yellowish color. It's in between, and it just, there it is. It's almost directly at the point now of this pin. So that's an
There it is.
acantharian. It's a really, really neat organism, but I'm noticing that it is really different. Even though it's round, it has a different shape than a lot of the other things we're seeing. It's being bumped. It's not moving. So it's not moving on its own. I'm going to try switching out.
So those would be really great things to notice too. Like if you draw that acantharian, you can write what you notice. Is it swimming on its own? What's happening to it? What do you notice about the other organisms around it?
And so this slide is going to be a lot less busy that I'm putting together now. I like to start with that broad overview, like the landscapeito, where you're seeing so much of so many different types of things. So the next slide I'm putting up holds much less liquid. So I'm cleaning the cover slip right now so that I can zoom in. So right now we're still at 40 times magnification, and that's great. That's like that broad view of the wetlands. And then now, there we go. I'm going to trade out this really busy slide and set it to the side. Here we go. Now, there's a lot more dark. It's going to take me a minute here to focus in because the slide itself is a different thickness. So where I focus, there we go. This is going to give us a slightly different perspective on what's in the plankton. I'm going to move my other slide to the side. So if you have any questions, feel free to pop them in chat. I'm going to move around a little while I'm noticing there's still a lot of those golden circles, which are diatoms. There's a lot less movement.
Janai, did you say that this has fewer drops of the concentrated plankton water?
Yeah. So this is about 3 drops.
Okay.
Just I've like stepped over to the side of the wetlands where there's like maybe a little inlet and there's a lot fewer birds to watch. So there's a lot less here. I can focus in more quietly on different things, but having that first look really gives me an idea of what's going on in the bigger picture. And so there's something that's moving here. It's moving gently in a circle towards the center. It's not golden brown. It's not a circle. Its shape is different. Mm-hmm. So I'm intentionally asking questions because this is how I started. I started by looking and wondering. If you want to get more into the weeds on figuring out what everything is, I do stream on Twitch and I post on Instagram, and we can dig really deep into whatever I know and other experts who come and visit what they know. But for right now, I just want you to look and to think about what you're seeing, because we don't have to know what everything is to appreciate it. I'm gonna switch. We're gonna zoom in right now because I'm really excited about that slow spinning organism in the center. So for me, that's something that I want to see closer. So now we're looking at this at 100 times magnification, and I can see a little more detail on it. It's coming in a little bit more clearly. And then while I'm noticing that, I'm also noticing there's this golden brown, it's getting bumped and twirled. See, there's a worm on the right side of the screen. Let's see, trying to get it in focus. Even though this is 3 drops, there's a lot of space for these organisms to move around in. You can see they're not squished. They have space. And so there's this worm. You can see those, it's got bristly spines sticking out from it. It has golden brown, I'm noticing, inside of it. So it's a zooplankton, the big category that it would be grouped in. It's an animal plankton, and it eats things that are golden brown. So that to me is showing how it eats the things that are golden brown on this slide here, and in the plankton are the phytoplankton. So it's giving me clues. Every once in a while I can actually see them eat. We'll see if this one, when it's using these little fine cilia or bristles around its mouth to create a current. See how it starts moving things when it gets close to them? Even if it's not very, very close, it still can, oh, no, it didn't eat that one. Sometimes it does. Sometimes I'll get to see them actually eat one of those diatoms.
So yeah, describing this movement in your journal would be perfect too, like exactly what Janai is saying. And if you have any other noticings and wonderings, you know, put that in your journal, because that's going to help us observe it more carefully. Like I notice how it's like doing like little curl ups.
Yeah. And when does it do those curl ups? Does it just curl up rhythmically all the time, or is there something that's happening that is causing that curl up, right? What does it do with the spines and the bristles on its body when it curls up? I'm trying to keep it in focus. It's, even though there's not much on the slide and only 3 drops of water, there's a lot of space for it to do its thing. So I'm gonna let it go now. And I'm gonna see what else there is here on this slide. So as far as the golden brown things, whoa, sorry. Trying to, here we go. These guys tend to hold more still. There's that one. It's not golden brown and it's moving slowly in circles, right? So I know, because I've looked up this organism, I know it's a dinoflagellate. And I'm part of what clues me into what it is it has this slow swimming motion. So it's moving. It's right now, it's just moving in a circle, but they can move in any direction. I notice it has this kind of a groove that splits it in half. So one of the things I like to think of with this one, it's like a party hat on top of a pair of pants, right? So it's got that kind of cone-shaped top. It reminds me of that, right? Having that, those 2 shapes squashed together with like a belt in between. And because it's holding so still, I'm gonna zoom in one time closer. These guys, here we go, now I have to refocus. These guys are really, really neat. I found them so fascinating. So they have one flagella, which is like a whipping tail that wraps around that groove. And then they have one that comes out between the 2 points. And I don't have it set up so we can see that very clearly right now. We can try going in closer if it cooperates. But
what magnification is this one?
Good question. So I just zoomed in again. So now we're looking at this 200 times actual size.
Cool.
Now, because I noticed that it has that waistband and it has those 2 points that groove down below, there's a golden brown thing just up to the left of it that seems to have a waistband and then a faint, like rounded points at the bottom. So it's echoing that shape. I'm noticing that it's not it's not perfectly round like these other diatoms. You can see those round golden brown circles have splotches of darker golden brown inside them, and they're really not moving. This other one here, it's like it's got a belt on. Right? It reminds me of someone with a belt around their waist, you know, with a little bit of roundness above and below. So here's the question: if you want to see this dinoflagellate, it's behaving so well. Right now, what we're doing is we're looking at this in dark field, and dark field is where I block the light coming through the bottom, and I'm lighting it from the sides. And the filter on my microscope that does that only works up to about this level of magnification, up to about 200 times magnified. But I want to zoom in closer. And so the way I do that is I go to something called bright field. And that means I take that dark filter, that dark black background away, and I let the light come up through. And I don't have the same type of color because the light's going to come through it. So I'm going to try that because this one's behaving so well. We might actually be able to see the flagella moving, that whipping tail that comes out between those 2 points that look like a pair of pants to me. So I'm going to try that. It's going to flash, it's going to go bright, and then I'll focus back. Oh, and there's another, hold on, I'm zooming up. That's another dinoflagellate that's just like crashing through and zooming by, sorry. They move so quickly. Some of them, I can't keep them in focus, but this one's just being really, really cooperative. So it's like that bird that decides to come really close and stand there and clean its feathers for you. That's this dinoflagellate. So here we go. So this is bright field. This is the same magnification, and I'm gonna zoom in one more level. All right, and so now you can just make out, here, let me get it back towards the center of the screen. You can just make out that really thin, maybe you can, that really thin flagella. I'm gonna play with the lighting to try and change the contrast a little for the bright field to see if I can get that. You can see that flagella wiggling right down between those bottom 2 points. So you can also see just barely the flagella that wraps around that waistband between the party hat and the pair of pants. This is a really neat one. So it's got, you can actually see there's like these lines and a pattern in the way, like the veins on a leaf, kind of. That it's coating, what it's made out of, that outer coating that holds its stuff inside. You can see where they kind of, where these plates come together and make a pattern.
These are really great. It reminds me of, I'm sure everyone can come up with some really cool, it reminds me of, because we're looking at some really unusual shapes.
Yeah, it's not your traditional shape. So, but you can see, you can just make out, oh, it flipped over for us.
Yeah.
That was neat. But you can just make out that flagella. So it is a dinoflagellate, and you can just barely see, I hope, that tail that's whipping out.
It's like a bird's beak, that opening. Oh, you can see the structure better when it spins around.
Yeah, it's being so cooperative, right? They're not usually this cooperative. Either they're super still and they won't move, or they move so fast I can't keep them in focus. So for me, this is a treat to be able to see it twirling so gently. And one of the reasons why I wanted to stop on this one in particular is because with bioluminescence, a lot of the bioluminescence that we see along the California coast is caused by dinoflagellates. I just wanted to see if this other one had its flagella out, and it doesn't. Let's see. Oh, it's going off the screen. So while we're here, those golden brown circles, those are diatoms. They come, well, as we scoodle around this slide, we'll see a lot of different shapes. So this particular one is a centric diatom. One thing I notice about this diatom is it has symmetry. So you could fold it any way around that center of that circle, and it's even all the way around. So we have, as humans, generally bilateral symmetry. So our left is like our right hand side. And this dinoflagellate, you know, it has that same type of left-right kind of symmetry. But there are others that we're gonna hopefully find on this little adventure. I'm gonna zoom out in a sec and we'll find some more that don't have that same type of symmetry, or they have different kinds of symmetry.
Before you zoom out, Janai, can you, to get a sense of like how big this is, if the frame is, I mean, if the,
yes, good question. Where's my little, I have a cheat sheet somewhere that I wrote all of these down on, because I usually have it on my microscope. It's approximately 0.5 millimeter across now, I think, is where we are. Wow. This is 400 times magnified. So we started at 40, and now we're at 400.
Okay, so 400, and so in the frame of the screen is 1 millimeter. Is that right?
Yeah, about 1.5.
Okay, so we're gonna do some math. We're gonna do some estimating. So if you're looking at your screen, it's about 1.5 millimeters. And how many of these little guys fit your screen? Can you estimate? 1, 2, 3, 4. You say maybe 5 of them go across? Yeah. So then we're taking 1.5 millimeters.
Just 0.5 millimeters, sorry. That was my bad.
Oh, 0.5 millimeters, okay.
Yep.
0.5 millimeters, okay. For the whole field of view.
Yep.
And 5, what did you call this guy?
It's a dinoflagellate. Oops, it just swim off, sorry.
So 5 dinoflagellates go across 0.5 millimeters. Okay, just trying to get a sense of scale. Thank you.
Yeah, no, it's a really hard thing, and I have a cheat sheet I just had to find to confirm, because so I'm really, really bad at numbers, and it's something that I struggle with in my journal, getting the data and the numbers and the measurements. So I appreciate you checking in about that, and I did have to look it up on my cheat sheet. Because I have a special slide that I used to measure the field of view, because I'm using a camera and I don't have a measurement. It
was huge.
It was huge. Yeah, that was a copepod, and they move too fast. They're like really quick hummingbird kind of. They're like hold still and then they zoom. So I'm gonna zoom out, and we're gonna go back to dark field, and we're gonna scan around. So I'm going to go right the way back to just the 40 times magnification. And I'm going to go back to dark field. So here we go. Give me a sec there. And we're going to, so we see there's that polychaete. So this white line, that's the edge of the piece of glass where I have the sample. And my cover slip, right, the part the glass that covers what I'm looking at, is 22 millimeters by 22 millimeters. It's a pretty small area if you think about it, less than an inch square. So, let me put my glasses back on so I can see where I'm scanning. So I'm noticing right over here, hold on, there are some interesting shapes. They're being bumped by the copepods, but there's like a little tiny half circle that I'm noticing right there.
The one that looks like a
That
horseshoe?
little copepod just swam by it, and it's pretty faint. If I turn up the brightness, right, I can see it, but it's really hard to see. So I'm going to try and zoom in on that. I'm really excited about some of the phytoplankton is great because it holds still most of the time. And it does have that golden brown color. So now we're at 100 times actual size. So the whole width of the screen, let me see if I can get it back, is about 2.2 millimeters. Let's see. So that's kind of a neat shape. I'm noticing that it's not one solid shape. It looks like it's a grouping. And one of the things that I know about phytoplankton is that they'll often chain together in the plankton. So I'm going to zoom in one closer. So we're going to go into 200 times here to try and get a little more detail on this. I'm going to try and focus. So you can kind of see there's little clumps of that greenish golden brown color in each of these. They look like little compartments or little pillows or purses that are linked up. I'm noticing that that I can't get the whole thing in focus at once. So I can focus on the right hand end a little bit, and the left hand end is out of focus. And with the microscope, what that's telling me is it's not flat. It's not in the same plane. So one end is closer to me when I focus here, and then I can change my focus and I can focus on the other end. But I can't really get them both in focus at the same time. So it's telling me something about the shape.
Yeah.
And it's getting bumped by copepods. But that idea that when you're looking at something through a microscope, we lose some of the dimensionality, and we have to think about how we're focusing on things to be able to understand what we're seeing. So it tells me, since it's kind of a circle shape and I can't get both ends in focus at the same time, that it's more like a spiral than a flat circle kind of coming at me. And then because this is it's a really neat one, I really like, there's like a clumping pattern to the golden brown green bits towards the center of each of those segments. They're actually cells, individual cells. I can try zooming in one more time if these copepods stop photobombing here. Those are those little zippy zooplankton. We'll go back to those in a sec. But I'm gonna switch out once, just because it's such an interesting shape and it's something to notice here. I'm gonna go to bright field, and we're going to go up to 400 times magnification. So here we go. Turn the brightness down a little. So now I can see those little, let me see if I can get it back towards the center here. Trying to play with the contrast a little. So one of the things I have to do is I have to fight with my camera as well as my microscope to get the image to look the way I want, to be able to learn about it. So right now I'm battling a little bit with my camera, but you can see here that I've just got that top right hand point in focus. You can see it's clear. And somebody's calling. It's okay. I have other family members that'll answer.
It looks like
Maybe.
in the little cells.
Private call.
Sorry about that. So there's kind of a pattern to the way those globules are kind of grouping around the center. Now I'm going to focus on that back point. So here's that back point coming into focus. Now I can get that more in focus. It's still the same shape, right? And they meet up with each other where the cells meet up. It's a little bit brighter. It's just it's a really interesting thing to see the way all of these cells here are chained together. And see, there's one in the center of this ring where it doesn't have anything. So I'm wondering, is that one's not alive anymore because it doesn't have, oops, there's a copepod real quick, photobombing copepod. It doesn't have that golden goodness in it, right? It's different from the others, right? But it's still connected. Is there something about the way, why is that one empty and the other ones have so much stuff in it? Is it, these are just questions that I put in my journal. These are the kinds of things I ask. Is it because the ends are the part that is dividing and growing and adding on to it? And so that's like, maybe that's the oldest one on this group and it's been dividing out from the sides. I don't, or has it been dividing from one end going around and maybe something just ate that one and not the others? I don't know, but there's so many questions just about this like little ring of 5, 6, 7, 8 cells.
So I
To
just
me
want
is
to, Janai, I just want to jump in here. That, you know, what you're doing is awesome. And I want to encourage everyone to not worry about what this is called and just to notice what Janai is walking us through, like her process of observation. Because this is really what's important in nature journaling, about like really observing and getting your creative juices flowing and asking those questions. You know, like, what is it? No, gosh, like, why is it doing that? What could that possibly be? Because oftentimes in our culture, we want to know the name, and that gives us a sense of security. And when we know the name, we feel like, oh, okay, now I can breathe. You know, we have Google, so we can like Google it on our phone and find out the name of everything. But really, what I'm encouraging people to do is just to sit with not knowing. And oftentimes in science, there are things that we don't know what they are, why they do things, where they go, where they live, how they reproduce, and put those in your journal, because that's going to help you become more observant, like a naturalist and a scientist. So just, I'm encouraging you to be okay with not knowing. Put all of those questions in your journal. Write everything you notice about that really weird little horseshoe shaped thing that we were looking at, because with identification, especially with birds, I'm a bird watcher. If I can't identify a bird, I write down everything I notice. What is it doing? How is it moving? What color does it have? What is its beak shaped like? And then I can go back later to a field guide and then I can look it up. But if I pulled out a field guide and started looking on my phone and making phone calls while the bird was in front of me, I'm going to miss that moment and the bird's going to fly away. So with nature, I really am encouraging everyone to just sit with just soak in what Janai is sharing with us. This is just an incredible part of life that's invisible to most of us. I mean, we could probably drink this, right? And we just never know what kind of beauty and structure is in it. So there's a lot of questions, and I think that maybe at the end we can share, Janai, can we share your chart with the
oh yeah, definitely.
We can put that on my website, and then so folks can go to that as a reference. But really, I want to encourage everyone to just be in the moment, draw what you see, write down all the words, the descriptions. And Janai did this beautiful way of like walking us through how she observes nature. And she looks at plankton way more than we do, right? So even people who know a lot about one subject are really great naturalists because they keep asking questions. And sometimes the answers to those questions will come right away, and sometimes they'll take a while. But I think that if you can sit with the not knowing and observe more, and if you discover the answer in your observations, that you're going to remember that. and it's going to be empowering, and it's going to be part of you. So it'll connect you to the plankton. So I just want to encourage everyone to, just write everything, that the questions that are coming up, or even if it's someone else's question that's come up, put it in your journal, and then we can revisit those questions later.
Yeah. And then I'm because I know we're kind of running well, for me it feels like we're running short on time. I would do this for 6 hours and just like.
Exactly. And we put your Twitch channel in there, and then we'll put it up again later, so that folks, if they want to visit with you live and looking at more plankton samples, they can do that with you and check out your Twitch videos as well. So while we've got you live, we've got about 10 minutes. Let's keep exploring.
Okay, so we've been looking at the phytoplankton. We've been looking at the dinoflagellates that are responsible. A lot of dinoflagellates bioluminesce, so that's a whole other thing. So we were looking at some dinoflagellates. And now in the center of the screen, this is a baby snail. It's a baby mollusk. It has this kind of curved shell. I'm noticing that it's kind of that snail shape. And we've looked at things with dark field, so the background is dark. We've looked at things in bright field, so the background is bright, and you see things differently when you look with different types of lighting. So right now we're at 40 times magnification. So this is as far out as I can zoom on this. And I wanted to share one other way of seeing plankton, because it's really striking to me. I'm gonna zoom in quickly to look at this at 100 times. So here we're going to zoom in 1, right, to 100 times magnification. I notice how it's got that curl already. I can see there's a little bit of movement inside of it. This one isn't swimming around. It's being really cooperative right now, holding still on this slide. I'm going to zoom in one more time. There's a lot of roundness to this organism. So I can't get the whole thing flat and in focus at the same time. So I see there's some golden brown in it. That's probably something it's eaten, because the other things in the slide are golden brown. But I can't get it all into focus in the same moment, because it's 3-dimensional. It's not flat, and it's thick enough that it won't let me. And before I switch out to what I want to share, there's a piece of a diatom on the right. It looks kind of like a piece of crystal with holes in it. And that's because diatoms, right, like those round ones, have a structure made of silica. And it makes this part of its what holds it together as a cell is made out of this silica, and they're golden brown when they're filled with life. And then this one here is just a piece of one that we can see that pattern in. It's not a living piece of a cell anymore. So what I wanted to show, I'm going to zoom back out now. There's one other way that I can look at this with my microscope, is by using something called a polarizing filter. And people who do photography probably know about polarizing filters, because they've used them on their cameras to help with the glare on the light. So I'm going to take out my dark field filter. It's going to go bright, and then I'm going to put in. I have just a little hacked together polarizing filter. So that one isn't doing a whole lot, but I have another filter. So this one I'm putting in above the slide, and it's at a right angle, and it changes the way everything looks. Let me see if I can just adjust this a little bit and drop it down. So now that snail is glowing. Do you see how the diatoms that were really bright or really dark depending aren't as shiny now? But that baby snail is literally like off the charts glowing. I have to turn the brightness down, and it still glows. So I'm going to zoom in 1. So this is 40 times magnification, and here we are. It's just so intensely bright. Look at that. Like the name for this property is bifringence. And it's not something that I know a lot about. But it is something that that I noticed when I was looking at plankton. Some things like the eyes on those copepods that were zipping by, like glow red. This organism here, this baby snail, has this amazing brightness that you don't normally see appear. Nothing is eating the is eating the snail at the moment. It's just holding still. What you're seeing is the curve and the brightness of the snail, and then part of its body is sticking out of the shell. And I'm gonna, let's see if we can zoom in one more, if it'll share anything else. So oops, nope, it won't. I can't. Sorry. That was too close with the filter on top. Do some of the plankton see the bifringence? No. So this is something that I'm doing to learn about it on the slide. This isn't something that's normally visible. So I'm using two different bits of polarizing film. But I just wanted to share that it's not just looking at it in one way. No, I did not squish the snail. It's still fine. In fact, here, I'll take the filter off. Sorry, I just turned on the chat. It's totally fine. What I did was I pushed on the film. So now it just looks like everything else, right? And what it's doing is it's moving its cilia here. Let's do this, and I'll put my other dark field back in. Here we go. It was just something that I really wanted to share because, here, brighten that up. So it's turned on its side. Because the bifringence is just another way, oops, there we go, to see. So it's rotated. It's why we're not seeing that curve from this direction. And it's right by a bubble. Oop. Let me go back. Here we go. I zoom out one more. So we're kind of looking at it from the bottom now, I think. There, did you see how it just got bumped? And now we can see it from a slightly different angle. Sometimes those copepods are helpful. I'm gonna zoom out one more. So now this was the original magnification we saw it at. Oop, and it turned back on its side, almost. I'm gonna go back to one. It's just about time to stop. So I'm gonna zoom in just one more here so you can see, right? It still has that curve of a shell. It's got these little cilia, these little waving hairs around the edge of it. But yeah, so there's an example of a larval plankton. It's only here for part of its life in the plankton. But that takes us to about 11 o'clock, and I know that Melinda, oh, sorry, last thing. Do you see that thing that looks, it reminds me of a sand dollar.
Ooh, yeah.
Kind of towards the center. So this is a type of diatom, and it's not got that perfect round symmetry. Let me see if I can just one more closer to it here. It's just a really special little one that I noticed. It's a single cell, but it reminds me of a sand dollar so much. It's just a really special little thing for me to end on, sorry.
Did you say this was a diatom?
It is, just like the golden brown one to the right of it, except this one's not alive. It doesn't have that golden color in it anymore. It's just that empty glass case. And anyone who wants to like dig into more plankton stuff, I'm happy to do that at another time. But this was such a broad introduction in a way that was designed, hopefully, for everybody to be able to observe and not be intimidated by the variety and alienness of what you're seeing. So
Yeah, this was awesome. And like someone else noticed, you can see the current that the zooplankton make when they swim by, and they just knock all these little particles left and right. They're getting jostled around, and you could see the flow. So it's pretty cool.
definitely.
It's pretty amazing that plankton is the base of our oceanic food web, which also includes humans if we're eating seafood, and our pets if they're eating fish meal. So, you know, without the phytoplankton in the ocean thriving, we would not have the life in the sea that we have right now. So this is like one of the most important things on our inner ocean that we don't often get a chance to see. So this is pretty cool.
So, and so we can do a couple little factoids at the end,
Yeah.
super quick. So if everybody just takes one deep breath, after all that trying to keep up with the plankton zooming around, hopefully you got to pick some out to sketch, and then another deep breath. So here's something to think about. The oxygen in that second breath was brought to you by phytoplankton. The phytoplankton in the ocean gives us half of the oxygen that we breathe on the planet. And that's a conservative estimate. So that's just one of those now that we've dove in and had a closer look at some of these small golden, brown, green organisms, realizing that they're responsible for so much of the oxygen on the planet, just wows me every time. So that was my my closing bit.
Thank you so much, Janai. I am going to, let's see, I think I had a slide here of, oh, I thought I did. Hold on one second.
There we go.
Okay, so we have some links for Janai's Twitter and Instagram. You can check out some of the great photographs that she has. Join her in the evenings for live streaming. Oh, here, I think I have it. Oh, it's not.
Oh, yeah.
Here. Let's see if I can share this.
I put in the links, Melinda.
Oh, thank you.
Okay, great. And I'm gonna share my screen.
Oh, and did you want me to look at that at that chart? I don't know if I can bring that up right now.
That's
but I
okay.
can try.
What we could do is we can post that with a video, and then so when folks are watching the video, they'll have the chart to take a look at. So
Great.
yeah, so thank you so much for bringing your expertise and your passion and your joy of plankton, and perhaps you can come back again, because we'd love to just check out some more things and spend time on looking at some of the other creatures and seasonality. I'm sure they're different plankton at different time of the year, different seasons. Yeah, it would be really awesome to take a look at. So for those of you who are new, these Sunday sessions are brought to you free for you to join in, and it's supported by donations. So I spend my time putting lessons together and making this available for everyone. So if you are enjoying it and you want to show me that you want more of these free sessions, then and if you're able to, supporting me with a donation is really greatly appreciated. It really helps me, and it helps me feel like what I'm bringing to the world is of value. And by supporting Spark in Nature and my work, you're allowing other people who may not necessarily get a chance to have nature journaling to have that opportunity. So, you know, I'm partnering with different organizations in the community, and I'm bringing nature journaling out into the public.